Dear learners, my today's topic for the lecture video is fermenter. We are going to discuss in details about the design of the fermenter, the working procedure, the different control parameters and the applications of the fermenter. We start with the introduction part. What is a fermenter? Fermenter is a large vessel containing considerable quantities of nutrient media by maintaining favorable conditions. It is mainly used for carrying out the process of fermentation. The design and nature of the fermenter varies depending upon the type of fermentation to be carried out. Invariably, all the fermenters provided the following facilities for the process, such as contamination-free environment, specific temperature maintenance, maintenance of agitation and aeration, pH control, monitoring dissolved oxygen, ports for nutrient and reagent feeding, ports for inoculation and sampling, provide all aseptic conditions at the time of sample withdrawal and addition of inoculum, complete removal of broth from the tank and easy to clean. It should be designed in such a way that it consumes less power, have less evaporation and can be used for long periods of operation. Let's study about the different objectives of the fermenter. The fermenter is mainly used to carrying out the fermentation process. So, the objective will be related to the different production of the fermentative products. What are they? They are to produce the microbial biomass, to produce the microbial metabolites, to produce microbial enzymes and also to produce the different recombinant products. What is common in bioreactor and fermenter? In both the fermenters and bioreactors, the fermentation is carried out, and fermentation in the anaerobic conversion of the sugar to carbon dioxide and alcohol by yeast. This is the common points between the bioreactor and fermenter. Many times we will come across that the term bioreactor is used instead of the fermenter word. So, do not get confused. These are the points which are common in a bioreactor and a fermenter. That is why they are used together sometimes. What are the different components of a fermenter? There are four main subdivisions of a standard bioreactor or the fermenter. Those are base unit, the culture vessel, the peripheral equipment and the instrumentation for measurement and control of key process parameters with links to supervisory of software and remote control facility. Okay. So, we will study in detail about the functions of the fermenter and the different types of fermenters available in the upcoming slides. This is the detailed diagram of a fermenter. You can see that it is having a motor which is driving the whole system. There is a acid base control for pH. This is the point. Steam for sterilization can be given out from this inlet. This is the inlet. Foam breaker liquid level is there. The propellers are there that are flat bladed impeller. Okay, we keep the culture broth here in this vessel. This is the culture vessel. This whole system is the culture vessel. There are baffles attached to the wall. This is the diffuser. This is the sterile air inlet and this is the harvesting or drain. From here we get the product. Okay, and this is the cooling jacket. Outside layer is the jacket for cooling, to cool the whole process. Okay. As you have already seen in the diagram, the different parts and their location in a fermenter. Here I am summarizing the different parts of the fermenter. Those are the materials that are used for the fermenter, the impeller setup, the baffles that are present there, the inoculation port, the sparger which allows the gas to get inside the whole vessel, sampling point, the pH control devices, the temperature control device and the foam controlling device. So, these three are the controlling device which are installed inside the fermenter and the bottom drainage system. What are the different peripheral parts and the accessories? Peristaltic pumps are there, medium feed pumps and reservoir bottles, the rotameter or gas supply and the sampling device. So, these four parts are the peripheral parts and the accessories that are attached along with the fermenters for its proper functioning. The different control measurements and the control systems of a fermenter are as follows. First is the speed control. This controls the speed at which the agitator will be moving and agitating the mixture of the fermenter. Then the temperature control, this is giving you the optimum temperature for the whole process to complete. The control of the gas supply for the supply of different types of gases that are required for the process of fermentation. The control of pH, whether the pH will be acidic or basic. The control of dissolved oxidation, whether it will be an aerobic process or anaerobic process, that can be controlled by the dissolved oxygen. Anti-foam control, the foam needs to be break down, and the feed control. Okay, so these are the major control system of a fermenter. There are several designs for a bioreactor or the fermenter. Those are stirred tanks. Okay, the stirred tanks, airlift reactors, bubble column, packed bed reactors, trickle bed reactors, and fluidized bed reactors. Okay, so here we will be discussing about them. First, we will discuss about the stirred tank reactor. In this, the mixing method is mechanical agitation. It requires very high input. The baffles are constructed within the build. So, these are the baffles. Okay, so these are the, here you can see these are the baffles. Applications include production of antibiotics and free immobilized enzymes. What is the drawback? That high shear forces may break the cells. Okay, this is the sparger attached. You can see it is quite similar to the normal fermenter design except the setup of the baffles. Okay, these are the flat bed turbine are present in this particular stirred tank reactor. The next design is the airlift reactor. Here the mixing method is airlift. Okay, so here you can see there is a upflowing stream. Okay, here you can see the upflowing stream is there and there is the downflowing stream. Okay, so this is how it is repeated like this in a circular movement. Homogenization of all the components are present and it is applicable in bacterial, animal, plant, fungi and yeast cells. Fine, inoculated gel beads are there. Internal tube, this is the internal tube, this is the central draft tube, this tube is the main equipment in the whole airlift reactors. Next is the bubble column reactor. In this the mixing method is gas sparging. Okay, so it has to have a sparger at the bottom which is allowing the air supply, the proper gas supply. The sparger is usually in the form of a ring which contains many holes in it, or it can be a single central small disc shaped thing with a single hole. Okay, it is a simple design, good heat and mass transfer rate takes place. It requires low energy input and gas liquid mass transfer coefficients depend largely on the bubble diameter and gas holdup. This is the exhaust point for the gas. Next is the packed bed reactor. Okay, so it has a column which is attached and it is also called as it contains the biofilm. Okay, it is mainly applicable with the biocatalyst. The pump is required to make fluid move through the packed bed. This is the packed bed column you can see. Okay, and this is the recirculated medium which is coming here. This is the stirrer, this is the sparger, this is the stirred vessel. Here you are getting the air supply and the gas exhaust point. So, it is mainly used in the wastewater treatment. This is the pump which is functioning for the whole fermenter reaction. Fluidized bed reactor. When the packed beds are operated in upflow mode, the bed expands at liquid flow rates due to upward motion of the particles. Here you can see that the gas is getting in from this point. This is the pump attached and this is the liquid is recycling like this. Again it is through the help of the pump it is going inside, and this is the gas exhaust and it is following a upward motion. Energy is required for this and this is mainly applicable for wastewater treatment. Trickle bed reactors. In this the liquid is sprayed onto the top of the packing and trickles down through the bed in small rivulets. Here you can see the small rivulets are present. It contains small pores. This is the liquid which is given from the top and it is entering from here, and this is the raw gas. This is the mineral nutrient and this is the washing water. This is the treated gas exhaust, and this is the filter bed with the different types of biofilm, this one. In the process, the gaseous pollutants on the surface of the carriers is adsorbed. So, it is a surface process and immediately biological mineralized by the microorganisms. What are the different applications of a fermenter? A bioreactor or a fermenter may also refer to a device or system that meant to grow cells or tissues in the context of the cell culture. These devices are being developed for the use in tissue engineering. It is also modular in nature and carry out all the processes of fermentation in a single contained environment. It plays a core role in the bioprocess. The stirred tank bioreactors are commonly used in fermentation industry. Due to simple technology and high yield, solid state bioreactors are widely used in industry. Ethanol fermentation is done by Saccharomyces cerevisiae in bioreactor. The organic acids example acetic acid, butyric acid are formed in bioreactor by the Eubacterium limosum. Thinamycin, an antibiotic, can also be produced in the fermenter or a bioreactor, and glucomylase is produced by Ariobasidium pullulans in bioreactor. So, this is a detailed application of the fermenter. With this, I end my presentation here. I hope the concept of fermenter is clear to you people. If you have any queries, you can mail me at the given mail ID, or you can also drop in a comment at the comment section. Follow my YouTube channel for more upcoming lecture videos related to the topic. Thank you.